And when it comes to quantitative analysis of really challenging compounds, it really is a game changer. This instrument combines absolute performance, efficiency, productivity and confidence to deliver the absolute power to quantitate even the most challenging compounds. When it comes to quantitative analysis of really challenging compounds, it really is a game changer. The Xevo TQ Absolute, this instrument is going to change the landscape of high performance tandem quadrupole mass spectrometry when it comes to sensitivity. The most sensitive tandem quadrupole instrument currently on the market, the Xevo TQ Absolute, allows users to achieve significantly lower limits of quantitation to meet their regulatory requirements, with up to 15 times more sensitivity for some challenging negative ionizing compounds than its predecessor. Next, let's take a look at efficiency. As a leading technology innovator in chromatography, mass spectrometry and thermal analysis instruments and software, we at Waters share a common mission with our customers: to leave the world a better place than we found it. Many of our customers are actively working to address environmental sustainability in balance with the need to continually reduce operational costs and improve efficiency. During the development of the Xevo TQ Absolute, we've addressed these challenges by providing an instrument with lower electricity consumption, gas consumption and heat output, lessening the need for air conditioning and lowering costs. As the smallest high-performance tandem quadrupole MS on the market, the Xevo TQ Absolute provides more analytical capability per square foot of lab space by minimising the LC-MS-MS footprint, enabling laboratory managers to fully optimise their facilities and increase efficiency. Productivity also goes hand in hand with efficiency, and in an ever-changing environment, flexibility can help protect against future unknowns. This is where our software comes in. The Xevo TQ Absolute is available on both our Waters Connect and MassLink software platforms, providing the ultimate flexibility to choose a future-proof solution to meet the requirements of your lab. Waters Connect is our newest platform for quantitation that provides a truly modern user experience. It is app-based, which ensures we can deliver superior efficiency and usability through apps such as MS Quan, which includes the unique exception-focused review functionality that can save up to 50% of the time spent on data review, ensuring your lab is as productive as possible. The Xevo TQ Absolute is also available on our tried and well-established MassLink software package, which ensures users have the versatility and flexibility needed for all application areas. And finally, confidence is key. We know that analytical laboratories face several challenges that are constantly changing, so it's important to have absolute confidence that your analytical instrumentation can keep pace. Essentially, the Xevo TQ Absolute is better by design. We've developed optimal probe positioning guidance that ensures users achieve the best possible performance while minimising contamination and reducing the need for inconvenient downtime to clean the instrument. A new source shield also maximises uptime by minimising contamination of the source from sample matrix or mobile phase salts. All these innovations combine to provide a solution that is truly game-changing. Now let's hear from Mark, who's going to talk to you more about specifically how the Xevo TQ Absolute delivers on its promise of absolute power.
As Kate said, one aspect our scientists are focused on with the development of the Xevo TQ Absolute is helping customers to achieve the lowest possible limits of quantification. When detecting ions in mass spectrometers, the ion must be made to create a measurable current. In other words, the incoming ion must be converted to a flow of electrons. Detecting positively charged ions is relatively simple because an electron is the opposite charge state to a positive ion. The positive ions are typically impacted onto a dynode held at a high negative potential to create secondary electrons, and then either an electron multiplier or photomultiplier can be used, which can be held at a relatively low positive potential to create a measurable signal. However, the detection of negative ions introduces complications because the negative ion is the same charge state as an electron. When a negative ion impacts on a dynode held at a high positive potential, it results in the generation of secondary electrons and/or secondary positive ions. If the secondary electrons are being used to create a current, the downstream device, for example an electron multiplier, must be held at an even higher positive potential. If the secondary positive ions are used, the downstream device can be held at a lower voltage, but the efficiency of the detector will be very poor for ions with low mass to charge. There are different approaches available to resolve this, but they have limitations either in terms of dynamic range or sensitivity for low mass negative ionizing species. Most detection systems use electron multipliers, but as the electron multiplier detector is not under vacuum, it requires regular gain adjustments and will eventually need replacement. The Xevo TQ Absolute and all Waters Tandem Quadrupole mass spectrometers use photomultipliers because they are permanently under vacuum and therefore do not require gain adjustment and typically last longer than the lifetime of the instrument. Additional complications can arise if the secondary electrons and secondary positive ions contribute to the generation of the current. The design of the Xevo TQ Absolute detector in negative mode means only the secondary electrons are detected, which helps provide a better representation of single ion events. This is required to help with trace detection of analytes, whilst maintaining the excellent dynamic range afforded by analog detection. To demonstrate this improvement in performance for low mass negative ionizing species, we can look at the anionic polar pesticides application. For these molecules, which have product ions with M/Z less than 200, there is an improvement of up to 15x relative to the Xevo TQ XS. In this back-to-back experiment, the same LC, chromatography and solvent standard samples were used. We analysed the samples on 2 Xevo TQ XS, 2 Xevo TQ Absolute and 1 competitor system. The results you can see are the ion responses relative to the Xevo TQ XS. For AMPA, glyphosate and glufosinate, the Xevo TQ Absolute detects more of the ions, and this means that trace levels of these compounds can be reliably and confidently detected and quantified. So to recap, we use a photomultiplier detection system to remove the need to adjust gain as the detector ages, and this system should last longer than the lifetime of the instrument. Our new improvements in the Xevo TQ Absolute enhance performance in negative ion mode, allowing you to obtain lower limits of quantification. All of this has been achieved while simultaneously reducing the footprint and keeping the low power and nitrogen consumption figures, which is what makes this instrument so remarkable as a high-performance tandem quadrupole mass spectrometer. But what does that mean from a real-world perspective? Well, it's over to Stuart now, who's going to take you through some data from key food and environmental applications.
So when we talk about the real world, and when I think about the real world, and when I think about food and the environment, because I've spent 15 odd years analysing for anionic polar pesticides, which are one of the most challenging set of negative ionization compounds to work with, I always think of my friend glyphosate, AMPA and glufosinate, and how over the years the analysis has in fact changed from one single residue method for glyphosate and glufosinate, and then other single residue methods to cover the other analytes that are now able to be covered in a multi-residue mix. So if you look at sort of the first image, I can show you glyphosate at 0.5 micrograms per kilogram in cucumber. Why cucumber? It's a universal matrix. So I managed to repeat this experiment many times, so it wasn't just a one and done. I can see it consistently at this level. But cucumber and glyphosate you wouldn't always expect to see. So when we start looking at the more heavy matrix samples such as cereals, we've got an example of glyphosate at 2 micrograms per kilogram and AMPA at 5 micrograms per kilogram in cereal in that heavy matrix. If you look down towards the bottom of the image, you can see the calibration graphs, and the linearity is important so we can get an accurate quantitation. When we think about the anionic polar pesticides, I'm not just thinking about glyphosate where I started 15 years ago. I'm thinking about the wider picture, the more multi-residue approach when we've got 10, 15 analytes in one separation method, in one extraction method. One of the particular challenges I remember facing was to analyse for glufosinate, including the 2 metabolites included in its residue definition. So if we look at the chromatograms, we can quite clearly see glufosinate and N-acetylglufosinate and MPPA all at 2 micrograms per kilogram. That gives us confidence that we can actually see the parent compound and the relevant metabolites that our customers are interested in. The all-important linear calibration is between 2 and 200 micrograms per kilogram. And again, that gives us the confidence that we're going to get accurate quantitation for this residue definition. So when I'm talking about accurate quantitation, we want stability within the analytical run. So when we're carrying out the measurements, I've got confidence, and that you've got confidence that what we're measuring is actually there, and we have got a stable signal. If you now look at the table, this is the limits of quantification for the 10 anionic polar pesticides that were investigated on the Xevo TQ Absolute. This is for the quantitation. This is for the confirmation of identity using the ion ratios, so that at the levels of quantification that I've quoted for 0.5 micrograms per kilogram in cucumber and 2 micrograms per kilogram in cereals except for ample, which is at 5, that is to detect the analyte, that is to quantify the analyte, and that is to confirm the identity of the analyte using the ion ratios. When I started glyphosate, we were struggling to get to 1 milligram per kilogram. When I left an operational lab and came to work for Waters 4 or 5 years ago, we were roughly getting to 0.1 milligrams per kilogram. Now, I'm standing here saying that we're actually getting to 2 micrograms per kilogram in a very heavy matrix in cereals, and we can get even lower in fruit and vegetables. This is just one way how the Xevo TQ Absolute is helping our customers get to those challenging reporting levels so that they can meet the needs of their customers. So I've talked about sort of food analysis and how the Xevo TQ Absolute is actually helping us with the challenging anionic polar pesticides. But if I step over into the environmental side of analysis, PFAS is one of the most challenging analysis that's currently labs are facing. PFAS, as one of my colleagues described it, it's an everywhere compound and it's a forever compound. We're increasingly finding more PFAS compounds that we should be analysing for, and we're increasingly finding that people are requesting that they want to go lower, they want to detect lower amounts of PFAS in their samples. How does the Xevo TQ Absolute help in this? Well, if you look at the C13 sulfonate, you can see we can detect down to sub-nanogram per litre levels. We could achieve this on the TQXS, but the major difference that we've got with the Xevo TQ Absolute is we're reducing down the injection volume. So we're moving away from that large volume injections of 30 microlitres, 40 microlitres, and coming back down to 10 microlitres. We've got a linear calibration range that we can calibrate over, which is again is ensuring that we can get the good quantitation that we require. One thing that we're also noticing is we're starting to see the branched chain isomers when we're actually doing this type of work as well in this system, just indicating that the system has got that bit more sensitivity to it. We're achieving the same results by putting less into the system, and we're seeing more. So whereas wastewater is important to see what's going into the environment, what's going into our sewer systems, drinking water is very important to monitor for PFAS as well, because that's coming into our bodies. So looking at C13 sulfonate, PFAS compound, again, we have got a similar level of performance in a drinking water sample. We've got good low levels of detection and low levels of quantification, and we have got that linear calibration range to ensure that we've got accurate quantification using the Xevo TQ Absolute. So to ensure that we can get consistently good results, I think one of my colleagues ran a series of 120 injections, and we monitored the performance of the method and the system over those 120 injections, which corresponds to roughly sort of 2 days analytical runtime in the system. And what we found was we were getting RSDs below 15% for the calculated concentration of the 20 odd PFAS compounds that we're monitoring. That's giving us confidence that we're getting good results that show good agreement inside what I might describe as a normal analytical run, and we have got extended data to indicate that the system is performing over a couple of days without user intervention. At this point, I could point out the fantastic limits of quantification we can get with the Xevo TQ Absolute, but we did something different. We actually took some wastewater samples and we took some tap water samples and we actually analysed them in the system to see what results we got back. And as you can see from the table, we've got a range of concentrations that we detected and quantified, and we've got some no detects indicating that on real life samples, we get results we'd expect to get. Using the enhanced sensitivity for negative ion and the Xevo TQ Absolute, I've given a couple of examples when we're analysing for PFAS in wastewater, PFAS in drinking water, where by reducing the injection volume, we're achieving equivalent sensitivity without having to do the large volume injections, which in itself has benefits all of its own. And that's me finished discussing some of the food and environmental data we have, and I'll now pass you over to my colleague Billy.
Thanks, Stuart. Another application area where the new Xevo TQ Absolute's performance will deliver real benefit is in small molecule bioanalysis. Quantitative small molecule bioanalysis is an application area that is extremely important in the pharmaceutical industry. From early stage discovery through to phase 3 clinical trials, without high sensitivity bioanalytical methods, the pharmacokinetic and pharmacodynamic properties of new drug molecules can't be determined. The Xevo TQ Absolute delivers the industry-leading levels of bioanalytical sensitivity required for these challenging applications. In a recent study, we quantified midazolam and imipramine, two examples of typical small molecule pharmaceutical compounds, in rat plasma using a simple protein precipitation protocol. With generic bioanalytical conditions, including a 5 centimetre long 2 millimetre ID, BEH, C18 UPLC chromatographic column, and water acetonitrile-based mobile phasers, we were able to detect down to a limit of quantification of 200 femtograms per mil for both compounds. Precision of 10% CV was achieved for both compounds at this level. That represents an on-column sensitivity of 250 attograms from diluted protein-precipitated rat plasma. This market-leading sensitivity, combined with its more streamlined footprint and lower operational running costs, makes the Xevo TQ Absolute a best-in-class instrument and a perfect tool for quantitative small molecule bioanalysis.
Thank you, Billy, Stuart and Mark, for providing some great insights into the capabilities of this truly game-changing instrument. From sensitivity and sustainability to efficiency, productivity and robustness, the Xevo TQ Absolute really does harness absolute power, enabling users to easily quantitate their most challenging compounds and make confident decisions fast. Thank you for joining us today. As always, our customers are at the centre of all of our innovations, and we'd love to hear what you think about this new launch, as well as take the opportunity to answer any questions you might have about the new Xevo TQ Absolute Tandem Quadrupole mass spectrometer. So please join us now for a live panel discussion. Hello everyone, and thank you for joining us for the Q&A session of today's launch event for the Xevo TQ Absolute. So I can see we've got some questions that have come in already. If any of you do have questions, please post them in the Q&A widget of the platform, and we can answer those in today's panel. So let's get started. So the first question we have here, I'm going to ask to you, Pete, and then maybe Stuart and Billy, you can provide your input as well. So, Pete, which applications do you see the Xevo TQ Absolute having the most impact?
It's really those applications where the high sensitivity is required. And when we think about sensitivity, to me, that means sort of flexibility. It enables us to make choices over what we want to do. We can obviously take advantage of that high sensitivity, but at the same time, it may be that we can bring other things into the method. So we could maybe inject less, we could maybe dilute more. So it really enables that choice to be made. So sensitivity for me is all about flexibility.
Stuart, what are your thoughts?
Thanks, Kate. It's sort of reinforcing what Pete said. It's about the additional flexibility. You can achieve the same levels of detection in the method by taking advantage of the sensitivity by putting less sample into the method, putting less sample into the system, or you can chase the ultimate sensitivity by maintaining your methods and getting the lower limits of quantification. For a compound such as PFAS, the limits of the quantification, they're only going down at the moment, and the TQ Absolute gives you the advantage of being able to go that much bit lower for those sort of types of analysis.
Fantastic. And Billy, sort of what applications from a biomedical research and pharma perspective do you see the Xevo TQ Absolute having the most impact?
The most impact is going to be on the more challenging compounds that ionise in negative mode. I think that's where you'll see the most impact. Again, as the guys have said, you're going to be able to use less matrix for the same results, so you're going to be able to protect your instrumentation more from a cleanliness point of view. You're also going to be able to get better precision at equivalent levels with increased sensitivity. In particular, biomedical research, where things like lipids, neg mode, ionising lipids, should have a really good impact on those kind of applications.
Fantastic, thank you. So we're staying with the sort of application side of things. We've heard from you, Pete, and Stuart and Billy from a F&E and a BMR and a pharma perspective. But moving over on to clinical, I can see we've got a couple of questions specifically around clinical and newborn screening, and whether that's a suitable application for the Xevo TQ Absolute, as well as any other clinical applications. So Mark, do you want to answer that one for me? Thank you.
Yes. So we've got a family of products, obviously, as well as the Xevo TQ Absolute. There are other Tandem Quadrupole instruments that we manufacture. And really for newborn screening, I'd recommend probably our mid-tier product, the Xevo TQS Micro, or possibly the Xevo TQD, both of which are IVD badged. And we also have a Renata DX system specifically for newborn screening. So those are more the kind of product range that I'd recommend for that particular application. The Xevo TQ Absolute would be, it would work for this, but they're probably more suitable products that we'd recommend for that. And David Ballantyne is our product manager for our IVD portfolio, so please do get in touch with David if you do want to discuss that further. For general clinical applications, really where we see that the power of the Xevo TQ Absolute is for things like steroids. So if you need a high sensitivity, tricky application like steroid analysis, so in POS mode, sort of testosterone, in negative mode, aldosterone, those are really the types of compound where we'd see the Xevo TQ Absolute being extremely important and useful.
Okay, great. I'll leave the mic with you for a moment, Mark, if that's okay. I think we've got quite a few sort of more product-related questions that I think you'll be able to answer really well. So there's quite a few questions relating to, I think, from customers that already have a Xevo TQ-XS. Obviously, the Xevo TQ Absolute is our newest high-performance product, but obviously quite a lot of customers already have a Xevo TQ-XS. So can customers with a Xevo TQ-XS run their TQ-XS methods on the Xevo TQ Absolute?
Yes, definitely. So we've got really good method transfer capability onto the Xevo TQ Absolute, and for the majority of applications, you'll probably just be able to switch it straight across the Xevo TQ Absolute and not make any adjustment. Now, obviously, in negative mode, the sensitivity has increased, and there will be some differences there in terms of maybe what's your most sensitive MRM transition now. So it might be worthwhile just double checking on those and checking that the fragment that was the most sensitive
or other Xevo instruments, can people use their existing ion sources from those instruments on the Xevo TQ Absolute?
Yes, definitely. So we've got the universal source architecture on all of our Xevo Tandem quads, and this transfers really nicely across onto the TQ Absolute. So I don't know if people are familiar with Unispray. That one has been extremely popular and gives a nice breadth of coverage for different application areas. You're generally going to see more sensitivity, more higher peak areas with that particular source. And then obviously for things, applications like nitrosamines where APCI is important, we offer that, or the full breadth of sources, APGC as well for any GCMS applications. So yeah, everything that you were able to use on the Xevo TQ XS, you can move across onto the Xevo TQ Absolute.
Fantastic, great. I think we'll pass over to the other side of the table for the next question. So I think I'll go over to you, Stuart, if that's okay. So there's a few questions here around PFAS analysis and that application. So the first one, are there any components of the Xevo TQ Absolute that need to be modified for PFAS analysis?
The modifications we make for the system, they're all based on the LC. So we've got a PFAS kit, which involves replacing some of the tubing in our Acuity systems to reduce the amount of PFAS that comes into the system from the LC unit components. For the Xevo TQ Absolute, there isn't any
actually, and it's obviously a lot easier to clean this source shield because it's a replaceable part. You can take it out of the instrument, put in your spare source shield while you're cleaning it, and you can just drop it into a beaker of solvent in an ultrasonic bath. So it's much simpler than trying to get your hands inside an awkward rectangular iron source and try and clean that enclosure by hand. In terms of the compatibility with other instruments, at this point in time it's only available with the Xevo TQ Absolute, but as with all technologies, we really see the future benefits of this, so we'll certainly look to see if we can make it compatible with other instruments in the future as well.
Okay, brilliant. And how fast is the Xevo TQ Absolute? So what are the maximum scanning speeds?
So it's the capabilities are the same as on the Xevo TQ XS in this respect. So we can have a maximum MS full scan mass speed of 20,000 Daltons per second. And then in MRM mode, for the quantitative analysis, we can go up to 500 MRMs per second. So that's a 1 millisecond dwell and 1 millisecond interscan delay. The one thing I would say is that just try to maximize performance by optimizing the dwell times and interscan delays. So the software will do this for you. But we do recommend, if you want the best quantitative performance, to maximise that duty cycle so that you're not scanning unnecessarily fast.
Okay. And so we talked a lot about the sensitivity improvements of the Xevo TQ Absolute, specifically for negative ions. What's the sensitivity like in positive ionisation mode?
Yes, so you'll notice that a lot of the application data that we've released of this product is focused in on negative mode because of the improvements we've made to the detector. But we still have extremely good performance in positive ion mode. And we haven't really focused in on that technology, but it's the Stepwave XS ion guide and the Z-spray source that really give us these improvements. So we have a two-stage ion guide, which is off-axis relative to the MS analyzer. So it allows the full encapsulation of the ion clouds generated in the ion source. And then neutrals and gas are vented from the ion source. Anything that, with a charge, so any ions are moved up to the sort of higher plane with a nice gradual gradient of voltage so that we're not causing any unnecessary fragmentation in source. And then when it's on axis relative to the MS analyzer, it's just fired straight in with really good sensitivity. So in positive ion mode, we get all of those benefits. And the application, there's an application note that's actually Billy has written for us on this for midazolam and imipramine, so small molecule pharma compounds in rat plasma. And he is able to see down to 200 femtograms per mil, so it's a really low level, and still getting a precision in the. He obviously was in with limited time at this point to conduct an experiment, so over 4 injections, which I know isn't a lot, but it's the only time that we had to do this experiment. He got a CV of less than 10%. So yeah, really good in positive mode as well as negative mode. Fantastic.
We've got another question that's come in for PFAS application, so we'll pass it back to Stuart before we come back to you, Mark, on some of the other questions. So question here for you, Stuart. Can the Xevo TQ Absolute be used for PFAS applications that utilise SPE cleanup too?
Yes, is a simple answer. Because it's the detector that we've made the improvements for, we can have several ways of exploiting the advantage using an SPE method. We can put less sample into the SPE cartridge and then achieve the same sort of limits of quantification in the method, or we can take advantage of the sensitivity by having a lower limit of quantification using the same SPE method and same MS methods. So that's sort of the 2 major ways where we can get the advantage. After all, it's a very sensitive negative ionization detector, so doing the cleanup just potentially allows the limits of quantification to be pushed even lower for PFAS.
Fantastic. So we'll just finish up back with you, Mark. We've got a couple more questions, so just again to remind everyone that if you do have questions, just post them in the Q&A widget now, because we've just got a couple more to answer, and then we'll be finishing the session unless there are any more questions. So these next couple of questions for you, Mark, around how people can get their hands on new Xevo TQ Absolute, which is great. We always love to help people with that. So the first one is around the cost. So can you tell people more about the cost of the Xevo TQ Absolute?
Yes, definitely. So the Xevo TQ Absolute obviously is our newest instrument. And if you already are a Waters customer and have a previous instrument, if you look on our website, we have this thing called the Flex Up program, and this would actually help you understand the benefits of trading in your previous product and getting a new Xevo top of the range premium product. If you're interested in any way, I would recommend speaking to your local sales representative, because obviously we're dealing with all these different currencies, so I can't give you one cost that would globally apply, but your local sales representative will be able to give you the price, and we're definitely in a position where we can offer an attractive price for this product.
Fantastic. And you mentioned the Flex Up program for customers who already have a Waters Xevo instrument. What about customers who might have competitor instruments but that perhaps want to maybe trade that in to get a Xevo TQ Absolute?
Yeah, so we've got a really extensive sales force who are very, very keen to speak to these customers as well. And if you've got some different technology, but maybe you're a bit concerned, or how can I transfer onto Waters, or you have questions, they're really, really experienced, will be able to answer your questions, or can pass it on to us. You could also contact me, Mark Roberts at waters.com, if you've got any questions around there. But we're also very happy to do some kind of trade-in program for a competitor's product as well. It's not a problem.
Okay, fantastic. So it doesn't look like we've got any more questions that have come in, but I do have a question for you, Gary. Obviously, you kicked off our video talking about the tandem quadrupole market and the mass spec market as a whole. We have been really heavily involved in the development of this new product, and we're all really excited about it. But could you tell me a bit about what excites you most about the new product?
Oh, wow.
There's so much, I know.
I guess, well, for us, I think we said it right at the start of the webinar. It's really exciting just to be able to help people solve problems, right? So when we get a technology that allows us to take a step forward, then you can see, okay, here's something we couldn't do before, a customer couldn't do before, or a problem we couldn't solve before. And I think the guys here have described some of those applications, right? Now you can do something that you couldn't do before on our previous products. And for us, working on that over the last couple of years, it's really exciting to see that come to fruition as it goes through research and development, and you see that technology starting to have an impact. And we work with our customers on beta testing and understanding whether or not it's working. It's quite an exciting process, obviously, for us to go through and see whether or not it's really going to bring something to life when we bring it to market. But really, it's kind of day one, right? We've just released the product last week. It's now out there in the labs for people to try. And so now the excitement really starts again. You get this new wave of people starting to look at the product and tell us, okay, this is what it's doing for me. So I can't wait for that, and I'm really looking forward to seeing the reaction from people trying the product.
Fantastic. Thanks so much, Gary. So it doesn't look like we've had any more questions that have come in. So I think with that, we'll finish today's session. So thank you all for joining us for the launch event and for this Q&A. If you do have any other questions, then please feel free to either get in touch with your sales rep, or obviously, Mark or myself, Kate Weirden, and we'll help you out and provide you any information you need about this new product. So thank you very much for joining us, and take care.